The majority of today's video is to introduce the AO1 knowledge on page 8 about systematic sampling methods. But before we start doing that, I wanted to take the opportunity to allow you to recap some of your abiotic and biotic factors knowledge. And I'm going to ask you to do this in relation to a sand dune ecosystem. Sand dune ecosystems are interesting for biologists because as you walk across them, the abiotic and biotic factors change significantly. And because of that, the abundance, so the number of individuals of each plant species, do change really dramatically as you walk across that ecosystem, which means it's a particular ecosystem that scientists do study using the systematic sampling method we're going to go on to talk about. Once we've done that little bit of a recap, we're going to focus on the systematic sampling method. We're going to start with an AO1, which is the compulsory practical that we will do, which is an investigation into the distribution of buttercups across a field, which I'm going to demonstrate to you. And then we'll think about how we apply systematic sampling methodologies to familiar and then weird AO3 examples in the exam. Okay, so a quick task for you. Okay, you might need to do a little bit of research, but if you could copy down the diagram of a sand dune ecosystem. So it begins at bare sand, and then as you move inland, you'll see that the landscape changes. It becomes sort of higher above sea level, and eventually we start to see trees and woodland. Okay, so what I would like you to do is to copy this diagram down and then to either from your own knowledge or a little bit of research, could you say how each of these different abiotic factors would change as you walk inland from the bare sand of the ecosystem to the woodland? And then join me back and I'll recap them. Okay, so wind speed. On the bare sand, there's very little plant cover, so there's no trees, just bare sand. So the wind speed is very strong, very fast. As you move inland and there are more plant species and eventually trees and woodland, then the wind speed decreases. Salt spray. So salt spray is something that would happen very close to the sea. So it's being high at the bare sand, but very low as you and getting lower as you move inland. Temperature would vary much more on the bare sand. So if you think about being on that bare sand, it's very, very hot when the sun's out because there's no shade, and very, very cold because there's nothing to keep heat. So fluctuates much greater. On bare land, those fluctuations become less dramatic as you move inland. The height of the land, well, that's increasing as you move inland. The organic matter in the soil, again, is increasing as you move inland because there'll be more plant species dying and recycling mineral ions, we have to be specific and name things like nitrates at air level, to the soil. The light intensity is at its highest, again on the bare sand. As you move inland, that light intensity decreases because there's plant species shading each other. The water content of the soil actually increases as you move inland, and that's because bare sand has nothing to retain the water and it drains away, whereas the root systems of the marron grass and the other plant species help to retain water in the soil, along with the organic matter, which helps to prevent the water draining straight through the substrate. Now we've got a soil forming. The one that is a bit strange is aspect. Okay, so this is sort of whether you're north or south facing. So that would vary depending on the side of the dune. So if the dune was the south side, then that would be warmer than the north side. Okay, what about then biotic factors. So here's a little picture of a sand dune. This is the Burr Sand, close to the sea, and as you move inland you can start to see grass species, and then later on these are succeeded by shrubs and eventually trees. So how do you think the biotic factors would change across the sand dune? Pause now and have a think. Okay, so if you join me back, biotic factors, of course, are the living factors. So if you've got an increase in plant species, you will have an increased number of niches and an increase in food sources for different animal species. So of course, there will be an increased number of animal species as you move inland, as there's more food sources available. That would mean that there's more competition between animal species, and also now we might have predation. But also, if we think about the plant species here on the Burr Sand, there are only xerophytes that are specially adapted to live there, so they don't really have any competition from other plant species. Whereas later on, you can see that there's lots of different types of plant species, and so much more overlap, causing competition between them. Okay, so that's a bit of a recap of abiotic and biotic factors. Moving on now to systematic samplings. This is on page 8 in your key notes. Now, it's not a very detailed methodology, so there's only really 3 mark points to remember, but there are different ways of performing a systematic sample. And so if the examiner presents an investigation to you, there might be slight variation on exactly how the sampling has taken place. So I'll talk you through it. So systematic sampling is about investigating the distribution of plant species. Okay, so this is across an area. So this is the bit that becomes really important. We only ever use this method in comparison to the random quadratic method when we want to see if the species present, or the number of species, or the number of individuals of each species changes as you move across an area. So one of the key ones is a sand dune system, because the abiotic and biotic factors change so much as you move across the area. The abundance of species, so the number of individuals of each species, and also the species present, does change. So this means that this methodology, the systematic sampling method, is used quite often when studying sand dunes. But there are many other examples that the examiners have used. So for example, they've had a riverbank. So moving from the side of a river and moving across the adjacent fields. And they've also, a very strange exam question one year, asked you to come up with a method to investigate the distribution of a particular plant species across a roundabout. So it's this across that we're looking out for to indicate that it's systematic sampling that we would pick rather than our random sampling method. So when we do a systematic sample, we use something called a transect. Now, it's a very posh word that means a line that dissects the area. Okay, the position of where we place that transect can be determined randomly, so we'll come back to that in a minute. But the important thing is that the transect is just a line across the area, and it's a line across the area at which samples are taken systematically. So we still use a quadrat, okay, but instead of placing that quadrat randomly within the sample area, we now, along that transect line, place our quadrat at fixed intervals, so systematic intervals. So these are predetermined intervals, for example, every 5 meters. Okay, so that is the main method that we use, and it's called an interrupted belt transect. Okay, so that's where we place our tape measure, transecting across the area we want to investigate, and then we place our quadrat at equally spaced intervals on one side of that line. And that's the one I'm going to demonstrate in the park in a moment. But so I would only ever describe that one if I was using it. However, the examiner might pick slightly different transect methods. So you can actually just place your tape measure across the area and record all the species that touch the tape measure, the line of the transect. You can also place two tape measures parallel to each other to create like a belt across the area being sampled. And then you can record all the species that are found between those two lines. The interrupted belt transect is quicker than both of those. Okay, so if the transect is placed randomly through the area, we just need to pick randomly our start point, if you like, to then take our tape measure out from. We should not just do one transect really across the area and count that as being reliable. If we wanted to make it more reliable, we would take more samples. Now in the keynotes, we've got something about running mean again. I'd actually get rid of that because I think it might overcomplicate the idea. So as long as you know that one transect really just tells us for one tape measure across that area, the distribution of species, it would be better science to then repeat that transect at different places in our sample area. And the more we do, the more reliable. So I might just change that there, instead of running mean, to that you would do many transects to make the data you collect more reliable. Okay, so that's our systematic sampling method. It's really simple, probably only ever 3 mark points on the exam. The same things can be utilised if you want more detail. So if you think about your quadrat area, you can give details like the size of the quadrat that's used and which edges are included and which are excluded to prevent the edging effect when you're counting your number of individuals. You can also use your other methods as well, your percentage cover and your percentage frequency, if that was appropriate to the species that you're investigating. So if we were looking at the systematically sampling moss, for example, so we wanted to see how the moss distribution changed across the transect, it may not be appropriate to count the moss species. Again, just like with our random sampling method, in our quadrat area for moss, a percentage cover would be more appropriate. Okay, so in our transect, when you place your tape along the area, you're investigating, record the species in your quadrat at regular intervals along the tape. So that's the basic methodology. Okay, so why use it? Well, this is only used if you want to see how populations change. Again, this is really important to look out for when it's changing across the area. So it might be that you're investigating the seashore, a sand dune, or moving up a mountain, so altitude's changing. So anything where the abiotic or biotic factors change significantly over an area, you may see a change in distribution of species across that environment. Okay, so here's the example of a seashore. Because the seashore changes so much as you move inland, this might be a suitable method for is a suitable method to investigate if the number of individuals of each population and also the species present, so which populations are present where in this seashore, change over the distance inland. So you'd start your transect here and when you tape measure up into inland and take your regular samples at intervals along that line. Okay, so this mark point is one that could be used, but I haven't seen it on the new spec at all. And it's just understanding that when you're doing your transect, so I want to investigate the changes in distribution of species as we move inland, I can find suitable sample sites and number them, but I might not have to investigate all of them. So I could use a random number generator to generate which of those sites I then run my actual transect from. As long as I do repeats, that increases reliability. Okay, so now I'm going to demonstrate in the park a transect investigation. So first, I lay out the tape measure from one side of the field to the other. I then place a 0.25 metre square quadrat at the 0 metre mark on the tape measure and counted the number of buttercups within the quadrat. This was then repeated at regular intervals of 5 metres along the same side of the line. It was possible for me then to repeat transects, okay, and I might have placed those repeated transects at random points along the field. Both of those would improve the reliability of my transect data. Okay, so let's have a look now at how we can apply this. But this, of course, is at A01. So this is a nice simple question. So I'd like you to have a go at this. So like I said, a sand dune system is quite a popular one for these styles of question, but don't assume if it's a sand dune, it's definitely going to be systematic sampling. It's not always. So what are you actually looking out for in the question to find out if this is a systematic sampling question? Well, you're looking for this idea of investigation of distribution from one side of the dune to the other. So in other words, across the system. Now we know it's going to be systematic sampling. So do you want to have a go at this question? For 3 marks. Describe how you could investigate the distribution of marron grass. This is grass species that grows on a sand dune from one side of the dune to the other. Okay, so you should have had a go at answering that on some paper in front of you. Let's go through. Okay, so the first mark point relates to laying the transect, the line, or the tape measure from one side of the dune to the other. Across the dune would also get you the mark point. The second mark point is then you would place quadrats at regular intervals along that line. Remember, it's on the same side of the line, and you might have to say a suggested interval, but in this case you didn't. What you can see straight away is if the examiner sets you up to answer about systematic sampling and you talk about random sampling and get mixed up and use the words transect but random sampling and random quadrats, then they will not give you your marks. That's why it's really important to be really clear. Final mark point is for what you do in your quadrats. So you can see they've given a range of different mark points here. You can either count all the marron grass plants within the quadrat area, or you can work out the percentage cover of marron grass within the quadrat area. You can use something called an abundance scale, which is not something that we've looked at, but is something that sometimes people use, particularly with shoreline ecology, so where people have done field courses going to the seashore. Or you can count the plants and record where they touch the line. So if you hadn't used the quadrats, but you had talked about having counting the plants and recording where they touch the line, or if you did a belt transect where you count the plants and record where they're between the 2 lines, you would still pick up 2 of the 3 available marks. That's why an interrupted belt transect is the best one to talk about. Therefore, you would pick up all of your mark points. Okay, fine. Okay, so finally, I just want to show you how you may be asked to apply your knowledge of systematic sampling to unknown practical examples. So in this exam question, the examiner introduced that there was a 75-metre tall tree releasing a very large number of small seeds. Ecologists use quadrats along a transect to measure the number of seeds at different distances from the tree. Their results are shown in the graph. So the question was, describe how ecologists would have used quadrats and a transect to obtain the data in the graph. Okay, so we're using those 3 mark points, but we're just applying them to this practical. So we'll get a mark point for saying that there was a transect line placed from the base of the tree trunk outwards for 160 metres, and that the ecologist would have placed a quadrat of known area, so a 0.25 metre square if you like quadrat, at regular intervals along that line. So the only difference then is what is going to be counted within the quadrat. So if you look at the data, it was the number of seeds. So it'd be the number of seeds that was counted. And because it's a mean, the examiner wanted you to recognise that that transect line was not the only transect line that was investigated, so that there were multiple transect lines from the base of the tree trunk out in different directions, and that the mean number would be the mean number at that position on the line. So the mean number at 0 meters of the number of repeated quadrats at 0 meters on the different transect lines, and the same for the 5 meter, 10 meter, etc. Okay, so that wraps up the introduction to our systematic sampling. It's not as well assessed as the random sampling methodologies you'll see as we work our way through exam questions, but it's important to keep it in mind whenever you're revising, so that it doesn't become something you forget about. Okay, so remember, systematic sampling is about investigating the distribution of species across an area. So that's the methodology we use there. And that's it for now.